This video will explore Chapter 4. Remember, Chapter 4 and Chapter 5 lecture notes will be covered on Exam 2. A study guide for Exam 2 will be posted in 2 weeks. By the end of this video, and subsequent review of Chapter 4, you should be able to discuss the learning objectives, using information from Chapter 4. The cell theory was developed from insights by Theodor Schwann, Matthias Schleiden, and Rudolf Virchow in the 19th century. However, it was Robert Hooke who in the 17th century coined the term cells by visualizing cells for the very first time. He did this using a crude microscope and a piece of cork. Additionally, prior to the development of the cell theory, Anton van Leeuwenhoek was the first to observe microorganisms using essentially a magnifying glass. The three tenets of the cell theory include the understanding that all living organisms are composed of cells, cells formed from pre-existing cells, and that cells represent the most basic level of life. Both animal cells and plant cells perform life-sustaining roles for biological beings. Cells also contain the four basic macromolecules, including both DNA and RNA, and produce and utilize ATP. This figure illustrates the diversity of cellular life on Earth. Due to the size of most cells in nature, microscopes are used to observe and study cellular characteristics. Two of the most common microscopes in biology labs are the light microscope and the electron microscope. The light microscope uses visible light to magnify and resolve biological images. The electron microscope uses electrons instead of light to produce an image. The terms magnification and resolution are important terms when dealing with the microscope, and terms you should know for Exam 2. Magnification refers to an increase in size of an image. Resolution is the ability of an optical instrument to show two objects as separate. Resolution also refers to the ability to distinguish fine detail, and focuses on the clarity of an image. Notice, in the image on the bottom of this slide, that in the best resolution option on the far right, you can easily see two clearly defined objects, separated by a minute distance. The next slide shows you biological images produced with both light and electron microscopes. This slide shows you the most commonly used units of measurement in the biological sciences. All cells, whether a prokaryote or eukaryote, contain a plasma membrane that surrounds the cytoplasm. The plasma, or cytoplasmic membrane, is a selectively permeable membrane, enclosing the cytoplasm of a cell. Again, the plasma membrane is selectively permeable. A selectively permeable membrane allows certain molecules and ions to move through the membrane, while restricting others. Unlike bacterial cells, the plasma membrane is the outermost layer in animal cells. The plasma membrane is made up of a phospholipid bilayer. Remember, an individual phospholipid molecule contains a hydrophilic, polar head, and a hydrophobic, nonpolar tail, as shown in this figure. You can clearly see that the phospholipid bilayer also contains transmembrane protein channels that allow certain molecules to enter and leave the cell. In addition to possession of a plasma membrane, all cells contain DNA molecules. DNA is the genetic material of the cell, and transmits genetic factors to subsequent generations. The cytoplasm, or cytosol, is a gel-like fluid in cells, protected by the plasma membrane, and contains molecules and structures important for cellular growth and development. In prokaryotic cells, the cytoplasm is everything inside the plasma membrane. In eukaryotes, the cytoplasm is everything inside the plasma membrane, and external to the nucleus. Look at the image on this slide, to examine the differences in cytoplasm for prokaryotes and eukaryotes. The cytoskeleton will be explained in detail in subsequent slides. However, at this point, you simply need to know that the cytoskeleton is an intracellular structure that supports the cell and provides an internal molecular transport system. On Earth, there are two major cell types, prokaryotes and eukaryotes. Prokaryotes are typically smaller and less complex when compared to eukaryotes. Eukaryotes contain organelles and a nucleus, prokaryotes do not. Eukaryotic organelles are small intracellular structures that carry out specific functions in the cell. Later on, in the chapter, we will explore individual eukaryotic organelles. In terms of the tree of life, prokaryotes are found in the domain bacteria and archaea. The eukaryotes are found in the domain eukarya. Again, the major difference between prokaryotes and eukaryotes is the absence or presence of a nucleus, respectively. Take the time to review this sample exam 2 question. If you have any questions about this slide, contact Dr. Flowers via email. Prokaryotes typically assume 3 main shapes: cocci, or spherical, bacillus, or rod-shaped, or irregular, or spiral-shaped. Prokaryotic DNA is located in a specific region in the cytoplasm, called the nucleoid region. DNA, RNA, and protein synthesis mechanisms are similar in prokaryotes and eukaryotes. Look at the image on the right to gain an understanding of typical prokaryotic structures and features. Both prokaryotic and eukaryotic cells contain precise metabolic machinery for ATP production and utilization. Also, some prokaryotes move with the aid of flagella. Moreover, some prokaryotes contain a pilus, which provides for the exchange of genetic material during bacterial mating events. Eukaryotic cells are distinguished from prokaryotic cells by the presence of a nucleus as well as membrane-bound organelles. In general, eukaryotes are larger than prokaryotes, although exceptions exist. Several experimental protocols allow biologists to separate intracellular components. The eukaryotic plasma membrane is fundamentally similar to the prokaryotic plasma membrane, in that it is a selectively permeable lipid bilayer. The major difference is the amount of proteins, carbohydrates, and other molecules embedded in the membrane. Also, eukaryotic plasma membranes contain sterols such as cholesterol, while prokaryotic plasma membranes do not. In animal cells, the plasma membrane is the outermost covering of the cells. Examine the figure showing an idealized animal cell and plant cell. Note the differences between eukaryotes and prokaryotes. The nucleus is a critical organelle in eukaryotes, and contains the DNA or genetic material of the cell. The nucleus is surrounded by a double membrane called the nuclear envelope. The nucleus also contains pores that allow for the passage of molecules, into, and out of the nucleus. This illustration examines the structure and components of the nucleus. The nucleus is able to import proteins into its nucleoplasm, due to the fact that certain proteins possess a nuclear localization signal, which is used to guide the protein into the nucleus. Chromatin and chromosomes represent different forms of DNA found in the nucleus. The nucleolus is also found in the nucleus. The nucleolus is the site of ribosomal RNA, or RNA synthesis. RNA is a critical component of ribosomes. Once formed, ribosomal subunits exit the nucleus, and contribute to fully functional ribosomes in the cytoplasm. This figure shows the nuclear envelope in more detail. Ribosomes are the sites of protein synthesis. Ribosomes are made up of two subunits. There is a small ribosomal subunit, and a large ribosomal subunit. Ribosomes can either be attached to the endoplasmic reticulum, or be free entities in the cytoplasm. The endomembrane system is a membranous component of the eukaryotic cell. The cytoplasm of the cell contains a system of membranous organelles that are suspended in it. The endomembrane system permits the transit of molecules throughout the cell. The nuclear envelope, Golgi, lysosomes, various vesicles, and the plasma membrane make up the endomembrane system. The endoplasmic reticulum refers to a network of membranes in the eukaryotic cytoplasm that connects the plasma membrane to the nuclear membrane. There are two types of endoplasmic reticula, smooth and rough As shown in the figure located on the slide, the rough endoplasmic reticulum is a type of endoplasmic reticulum that contains ribosomes attached to the membrane. The ribosomes are shown here as red balls. The rough endoplasmic reticulum plays a role in protein synthesis. Smooth endoplasmic reticulum, unlike rough endoplasmic reticulum, does not contain ribosomes attached to the surface. Smooth endoplasmic reticulum plays a role in lipid synthesis. The Golgi complex resembles a stack of pancakes as shown in the figure. The Golgi complex, Golgi bodies, or Golgi apparatus, is involved in the secretion of certain proteins. The Golgi complex is involved in the processing, packaging, and secretion of proteins. Exocytosis is a process by which the contents of a cell vesicle is released to the exterior, through fusion of the vesicle membrane with the cell membrane. Lysosomes are organelles in the cytoplasm of eukaryotic cells, containing degradative enzymes enclosed in a membrane. The hydrolytic enzymes are used to destroy microbes and other ingested biomolecules. Again, lysosomes are organelles containing digestive enzymes. The interior of lysosomes has a low pH that makes lysosomes acidic. The acidic nature of lysosomes contributes to the breakdown of ingested particulates and microorganisms. Lysosomes play a role in phagocytosis. Phagocytosis refers to the ingestion of solids by eukaryotic cells. The lack of specific enzymes in the lysosome can contribute to human disease. The process of phagocytosis includes 5 steps: chemotaxis, adherence, ingestion, digestion, and killing. This slide shows the movement of vesicles through the cytoplasm. Review the figure on this slide in order to understand how proteins are made and move within the cell. This slide shows the movement of various proteins through the cell. It shows how proteins are ultimately embedded in the membrane. Vesicles containing specific proteins bud off from various organelles, move through the cytoplasm, and fuse with specific membranes. You must know the 5-step process of vesicle budding and fusion illustrated in this figure for exam 2. The mitochondria is an organelle that contains the necessary enzymes for the Krebs cycle and electron transport chain. The mitochondria is responsible for producing energy in the form of ATP for the cell, via cellular respiration. Cellular respiration will be covered in detail in Chapter 7, and describes a series of redox reactions in a membrane that generates ATP, in which the final electron acceptor is usually an inorganic molecule, such as oxygen. The mitochondria contains two membranes, an inner membrane and an outer membrane. The inner mitochondrial membrane is the site of the electron transport chain. The inner mitochondrial membrane contains many folds, designed to increase the surface area of the organelle. The endosymbiotic theory is a model used to describe the origin of eukaryotic organelles. Moreover, it is a model for the evolution of eukaryotes, which states that organelles arose from prokaryotic cells living inside a host prokaryote. The cytoskeleton literally means cellular skeleton. Just like humans have a skeleton, individual cells also have intracellular skeletons. The cytoskeleton consists of microfilaments, intermediate filaments, and microtubules that provide support and movement for the eukaryotic cytoplasm. From this view of the cytoskeleton, you can see how the microfilaments, intermediate filaments, and microtubules provide a cellular support system, as well as a molecular transport system. Take the time to review this sample exam 2 question. If you have any questions about this slide, contact Dr. Flowers via email. Microtubules are one of 3 cytoskeletal structures, made up of the protein tubulin. Microtubules are hollow tubes, consisting of dimeric proteins. Microtubules help position organelles in the cell, move chromosomes, and serve as tracks, by which vesicles and other molecules are transported throughout the cell. Motor proteins, such as dyneins, kinesins, or myosins, are proteins that literally carry vesicular cargo to various parts of a cell, by walking on either microtubules and microfilaments. If you look at the picture of the motor protein on this slide, the protein domains facing down look like little feet that walk on cytoskeletal proteins. The protein portion facing up are somewhat like hands that attach and carry vesicles to particular cellular destinations. Intermediate filaments are one of three components of the cytoskeleton that are arranged in bundles and participate in the mechanical support of the cell. Microfilaments are one of three components of the cytoskeleton. Microfilaments are the smallest filaments of the cytoskeleton. Microfilaments have roles in cell movement, muscle contraction, and cell division. Microfilaments are composed of actin subunits. Flagella and cilia are two eukaryotic structures that play a role in cellular motility and nutrient acquisition. Flagella are whip-like structures, and cilia are short hairs that typically surround the cell. Flagella are composed of a 9+2 microtubule arrangement, as shown in this figure, of a cross-section of a flagellum. Take the time to review this sample exam 2 question. If you have any questions about this slide, contact Dr. Flowers via email. Chloroplasts, vacuoles, and cell walls are structures that are found in plant cells. Plastids are specialized structures in plants that perform specific functions. Plants contain chloroplasts, amyloplasts, and chromoplasts in their cytoplasm. Chloroplasts are chlorophyll-containing organelles that perform photosynthesis in plants and other eukaryotes. Chloroplasts possess an outer and inner membrane. The stroma is the liquid inside the chloroplast, where other structures such as the thylakoids reside. The central vacuole is an intracellular inclusion, surrounded by a plasma membrane, that serves as a storage organelle. The cell wall is the outer covering of plant cells, that provide support and protection for the cell. The cell wall of plants is mostly composed of cellulose. Cell adhesion molecules, cell junctions, and the extracellular matrix are important structures found in animal cells. Cell adhesion molecules are a group of transmembrane proteins, such as cadherins, integrins, and selectins. Cell adhesion molecules are cell surface proteins that bring about cell-to-cell adhesion by binding to molecules on the surface of other cells. Cell junctions are proteins that promote intercellular connections between the plasma membrane of adjacent cells of animal tissues. Cell junctions link cells to each other, various tissues, and regulate tissue homeostasis. Anchoring junctions, tight junctions, and gap junctions will be explored in the next few slides. Anchoring junctions attach the cytoskeleton of a cell to the matrix surrounding the cell, or to the cytoskeleton of an adjacent cell. A tight junction is a specialized connection of two adjacent animal cell membranes, such that the space usually lying between them is absent. Gap junctions are a type of cell junction, in which adjacent cells are connected through protein channels. These channels connect the cytoplasm of each cell, and allow molecules and ions to pass between them. The extracellular matrix is a suspension of proteins and carbohydrates secreted by cells and supports tissue growth, cell division, cellular locomotion, and other cellular functions. Glycoproteins are proteins that have sugars attached to them, and make up the extracellular matrix. This final slide illustrates cellular components of bacteria, plant cells, and animal cells.